My lab is largely interested in studying bioactive lipids, their effect in human physiology and disease, and developing technologies to study them in different mammalian and other systems. We all know lipids as cholesterol, triglycerides, and dietary lipids, but very little is known about bioactive lipids. These are very important lipids and they almost function like hormones and regulate different signaling events in humans and other organisms too. What we have been able to do over the past few years is map out how these lipids are either made or broken up and how these signal in different components of our body. For example, the brain, our immune system. And we now know that the levels of these lipids are very, very important in our body in different tissues. And even a little bit of change in these levels causes diseases. So now we're trying to figure out what are thresholds that we can tolerate levels of these lipids in our body and how their disruption might cause diseases and whether we can rectify this. When we started working on a class of bioactive lipids called lysophosphatidyl lipids, very little was known about its biology or its signaling events in different immune cells. What my lab did was we became the first lab to sort of synthesize these lipids. And we found that whatever we synthesized was able to do remarkable biology. And that sort of opened up huge opportunities for us to study this now with the synthesis capabilities that we had in lab. So that for us was a significant breakthrough moment. We have generated a lot of knowledge over the past 7-8 years on our understanding of lysophosphatidyl lipids. We found out enzymes that can make this. We found enzymes that can break this. We've also now found certain receptors that respond to these lipids. Now we know to some degree the extent of which all these enzymes and receptors are involved in different human diseases. The goal for us moving forward is whether we can now use this knowledge to develop therapy for different human neurological and autoimmune diseases. When I finished my PhD, I was invited to visit iiserpune by Professor Shashidhara, who was then at iiserpune. When I came here, I saw that iiserpune had fantastic infrastructure. It had a young set of faculty who were a lot like me, who had just come back from postdocs or PhDs abroad, and they're doing phenomenally well. So if they could do this in the current infrastructure, I thought I could as well. After visiting iiserpune, I also visited different institutions in India where I found the situation to be the same. And given that we had infrastructure, a lot of people coming back, a lot of very intelligent people in the system, I thought I should come back and, join the bandwagon and possibly push research areas in India. Another thing I found is when I came back after my postdoc, I found that there was very little sort of development in the field of lipidomics. And I thought, this can be a niche area for me and my lab, along with studying bioactive lipids. And, that helped us push our lab further in the next few years. To be honest, I don't have any formal biology training. I did my undergraduate work as a chemical technologist from Udacity, which is in Mumbai. I majored in pharmaceuticals and fine chemicals, but I did not have any courses in biology. I then went for my PhD largely to start off doing synthetic organic chemistry, but I ended up liking biochemistry. But again, here I had no formal sort of education in biology. And then for my postdoc, I sort of started studying physiology using chemical tools. And it's a combination of all these three things that are currently ongoing in my lab. So I started my lab in 2016, and we use principles of technology development, we use principles of synthetic organic chemistry, biochemistry, and physiology, and try to address a problem in a wholesome fashion. We develop a lot of technologies around mass spectrometry, which is a technique to measure mass of molecules. Now, we do this largely in the context of lipids, where we are measuring masses of different lipids, and based on that, we can quantify and measure different lipids. Now, when one develops a technology, the best use of technology is its ability to be able to be used in different sort of questions. So Perhaps my first collaborator is Roop Malik, who is a former Infosys Prize winner. And together with him, we have studied different problems, such as, understanding how lipids are transported by the liver or how the process of phagocytosis works, all of which are very interesting using technology developed by his lab and mine. Now, the advantages of collaborating is twofold. One is people get to try and test the technologies you have developed, and you get to try and test different technologies they have developed or different problems they are studying. And it's a synergy of these two that is very, very enjoyable. There is no substitution for hard work. There is no substitution for perseverance. Whenever anybody starts their own independent lab, the first 2 or 3 years are really hard. But they're probably the most enjoyable as well, because you're spending time at the bench, you're interacting with students, you're trying to develop a character for your lab. And my advice to younger people who are starting their lab is don't lose this enjoyment. While I was starting my lab, I faced several challenges. For example, I didn't have any sort of training post my bachelor's in India. So I didn't know how different bureaucratic processes, how the granting system and, other things in India work. But if you keep at it and you keep pushing yourself to do these things, things eventually work out in any setting that you are. When one starts to do research, we don't expect to win prizes. We do research because we love doing it. And this prize is really a recognition on both a national and international stage for the work done by the wonderful graduate students and other members in the lab. This is sort of a representation of their work and their efforts. And it does two things to us. Number one is it puts our research in the limelight. It shows people the importance of bioactive lipids. And second is it now sort of compels us to push this further and get it into real practice in the next few years.